Our final key speaker for today's event is Professor Warren Tate. He is an internationally renowned molecular biologist, and he's been at this university since 1975 as both a scientist and a teacher. Not quite as long as that building, but nearly. In fact, almost as long as the Neurological Foundation has existed, so it's fantastic. Part of the furniture. In 2010, he won New Zealand's Premier Science and Technology Honour, which is the Rutherford Medal. No mean feat. And in recognition of this huge contribution to health research, it was well presented. In this lecture, Professor Tate puts Alzheimer's disease under the microscope, and he looks at the role of proteins on the brain and the potential role they may play in treatments of the disease in the future. So, without further ado, please welcome Professor Warren Tate.
Thank you very much. I've been in Dunedin so long because I first came down as an honours student from Victoria University on a beautiful autumn day to go to St Margaret's College ball here, and just fell in love with Dunedin, and I haven't been able to leave ever since. So I'm a biochemist, molecular biologist, and I work on brain disorders, motivated by the importance of these for the individual and society as a whole in terms of health diseases, but also particularly motivated because I have family members and close family members affected by these disorders. Indeed, my father died of dementia, and today I'm going to be talking about Alzheimer's, and actually it was his birthday today, so I'd like to dedicate this lecture to his memory. Now, when we're, of course, doing research on such profound topics as I'm going to be talking about today, it's all done by a research team. So I'd like to start off by introducing you to my research team who work on Alzheimer's. First of all, the senior scientist, Dr. Margaret Ryan, and I'm going to talk about some of her work later in the talk. She works with me and also with Dr. Joanna Williams in anatomy. And then a really important person over here, Katie Bourne, who manages the production of the brain protein that I'll be talking about through the talk. A couple of students who've just now moved on to Australia to further their career development, someone next door who I don't know who that is. And then our 2013 group here, Jeremy, who's been with the group for a year, a new student, Josh. Kieran was a medical student that worked with the group over summer. We enjoyed having him here. And then a student from the University of Potsdam in Germany who's been with me for a few months, and it was her last day yesterday, and we shed a few tears mutually for her departure. And I'm very grateful to the contributions of this team that has allowed the kind of work that we've done to be accomplished. So what's important for me to know when I'm working on a brain disorder, and there are 4 or 5 key points. I want to know what the underlying mechanism is for the disease, and in lay terms, we can think that's the cause. I want to know whether there's any diagnostic biomarkers, and the lay term for that, is there a diagnostic test? I want to know if there's any potential therapeutic targets, and what that means is that are the targets that might actually lead to the development of drugs that can be used as a therapy against the disease. And critically important, and I'll build on this theme, is that we need to know whether there's an early or even a predictive diagnosis, so that one can know early enough that the disease is there to have a good chance of actually doing something to either reverse it or basically slow it down to help the individual. So these are important scientific questions, and these days, maybe because I'm in the age group where some of these diseases are critical, is that I like to feel that the research we're doing is connected to the needs of individuals with these brain disorders, and particularly where we might actually be able to get some help. Now, for Alzheimer's disease, which is the subject of my talk today, if we apply these kinds of ideas, we can ask, well, what about the molecular cause? And in fact, we know the gene that's actually responsible now for this illness, and we know quite a lot about the biology. We don't know everything, but we have a good handle on that. So for Alzheimer's disease, we can put quite a strong tick there. However, when we look at diagnosis, of course, the diagnosis of Alzheimer's tends to be when symptoms appear, when people are suffering from memory loss and confusion and whatever. These, in fact, are thought to be symptoms appearing at a rather late stage of the disease. In fact, it's thought that maybe the disease started 20 years before and has been slowly developing before these symptoms came out. So when you ask the question, do we have a predictive or early diagnosis, we can say this is not yet available and this is a real need. And then we can ask, what about drug therapies? Are the drug therapies available? Well, the cupboard is pretty bare. There's a group of drugs available which can, you could regard them as mild drugs. They can slow the onset of the illness maybe by 6 or 12 months in some patients. But there's plenty of gaps there for new drug therapies. But there's intensive research going on, and actually there's quite a lot of promise at the moment in the trials that are just about to be started, and I'll mention that. So our multidisciplinary research team at Otago, in fact, is focusing on these two things because we believe that it's important to have movement in both predictive or early diagnosis and available therapies, so that if you predict a disease is there, you can actually do something about it. And I'd just like to kind of expand on that by looking at a couple of other diseases that affect my own family, just to kind of, emphasize the importance. The top one there, MECFS, was classified as a brain disorder by the World Health Organization in 1992. I have close family members affected by that. And when we look at what do we know about this, there's an inferred cause, mostly likely to be a viral infection, but there's no diagnostic test, there's no therapy. And so for the estimated 20,000 New Zealanders who are affected by this, knowing that they've really got the disease and getting any help medically there's not any support there. So they are in a very difficult position. So in 2010, in fact, I've started working on this as well, with an emphasis on trying to develop a diagnostic test, but also with some concentration on what therapeutic opportunities are available. The other one, Huntington's disease, I have a family member, greater family member, who's affected by the disease. She's a contemporary of mine, and when she had learned that her mother had died of Huntington's disease, she knew that she had a 50% chance of getting the disease. But at that stage, we didn't know what the cause was. But in the mid-1980s, the cause was determined, the gene was identified, so we had a cause. Immediately, a predictive diagnostic test could be developed. And so she courageously took the test and found she had inherited the bad gene from her mother, and so therefore she was going to come down with the disease. She had no symptoms at the time. But there was no drug therapies available, so there was nothing that could help her. She was in a stage just waiting for the disease to develop, and she's now got the disease. And I feel rather guilty as a molecular scientist because although in the 1980s we we identified the protein that was affected, we still don't know actually what that protein does. So even, 30 or 40 years sort of afterwards. And I want to just give you a little bit more information about my family affected, and I'm not going to tell you family secrets here. This was the subject of a listener article in 2009, so I'll just tell you some details that were in the listener article. This is the daughter of Bridget, of the person that I was talking about before. So she had a 50% chance of, as a third generation, of inheriting the illness, and she had a child, and then wanting to have a second child, she decided she should take the test to determine whether she inherited the disease, and if so, take some steps to try and break the disease in the next generation. So she took the test, and she's also got the bad gene. So she is symptomless at the moment, but ultimately will develop the illness. So what she did, given that we didn't have any drug therapies available to push the boundaries back for the disease, she used reproductive technologies and molecular technologies, namely in vitro fertilization. So created some embryos in a dish, and you can tease an individual cell off those embryos, not affecting their subsequent development, and do molecular tests on them. And she did that, and pretty well all of the embryos had the bad gene. Fortunately, there was one that didn't, and that one developed into this child here. So the cycle is broken, but I think it emphasises to you, when you don't have a drug therapy, to go with an early diagnosis. After all, what's more predictive than being able to predict the disease in a small number of cell embryo? Had the predictive disease, but no therapies, and so, this is a major problem. And this is why it's so important to be working on the two things together. So, let's go back to Alzheimer's disease then. So what about Alzheimer's disease and the molecular cause? And so we know now that the particular protein, a gene encoding this protein, amyloid precursor protein, a bit of a mouthful, but we generally refer to it as APP, is the critical protein that's the causative agent, and I'll embellish that. So this is a protein which is stuck in a membrane here. This is a membrane there. And most of the protein actually extends outside the neurons in the brain into the extracellular space. But the critical, interesting thing about this is that there are sites on the protein, and listed here by these Greek symbols, beta, alpha, gamma, and epsilon, which are cutting sites. There are protein-cutting enzymes in the cell which can cut this protein, specific enzymes for each site that can cut the protein at that site. So how do we know that that's critical for Alzheimer's disease? Well, although most of us think of Alzheimer's as what we call sporadic Alzheimer's disease, which develop with age, there is a small group of people who have familial Alzheimer's disease. That is to say, they develop the disease early, and it's an inherited disease. And if we look then at some of the examples of people that have this inherited disease. So now I'm just looking at those sites there that I mentioned before, these protein cutting sites. And down here, it's a bit complicated, but these are the individual amino acid units of the protein through this region. Here are the cutting sites. You'll hear about this enzyme again, base 1, the beta site, the alpha site, and the gamma and epsilon sites there. And the thing I just want you to notice is that there are mutations that have been identified close to these cutting sites. The sweetest mutation here, close to this site here, and that means the efficiency of cutting is greater, and that leads to Alzheimer's disease. A number of mutations here, and you can see they're named from where they were discovered, Italian, Dutch, Iowa, Arctic, Flemish, this one Swedish, and a number over at this cutting site. So that was the compelling evidence that told us that this gene and this protein was the important protein. Just recently, last year, there was a study in the Icelandic population. They've got a big genetic study going on called DECODE, and so they looked at 1,800 older people, looked at their medical histories and actually their gene sequences, and discovered, in fact, there was a group of them which were free of any Alzheimer's type dementia, who had a mutation down close to this cutting site. So it seemed like that in these people there was a protective mutation. Probably the cutting is of lower efficiency, and we know then from that that actually protects against mutation. So this the gene for this is on chromosome 21. We have 2 copies of that, and even if you only have 1 copy of this altered gene, you have almost 10-fold less chance of actually developing a dementia type illness as Alzheimer's. So we know that's a protective. Okay. So if we then focus on these sites here, what we know is that there are 2 pathways. One pathway, the minor pathway, produces a peptide called amyloid beta, and if that's produced in relatively low amounts, it does, we think, normal, important things for the brain, keeps it physiologically healthy. But if this pathway becomes stronger and we produce too much of this, then this becomes neurotoxic, and it's this that we think is responsible for the development of Alzheimer's disease, and I'll give you a little bit more detail about that in a moment. The dominant pathway here actually produces a larger protein fragment, which we call its secreted amyloid precursor protein alpha, or SAP-alpha. And the important thing about that is that that's neuroprotective, and this is the one that we've concentrated on in my research lab, and I'm going to talk to you about. And I've called this the bad guy and this the good guy, just to give us sort of some anchor points as we go through the rest of the talk. So what we think then is happening is that normally in a healthy brain we have a balance between these 2 pathways, and so if you like, the seesaw is kind of horizontal when it's balanced. But if the beta amyloid pathway is elevated, then it becomes out of balance, becomes too heavy. So the seesaw, if you like, is weighed down, and there's a bit of expense for the other pathway, the major pathway, because that will be a bit lighter. So these 2 molecules are clearly interrelated by the fact we have these 2 pathways cutting up the APP, and that's important for the development of the disease. So you could say, well, which molecule should we focus on? Should we focus on the bad guy, the beta amyloid? And I have to say that most scientists around the world and individual companies, that is their focus. They're solely focused on that bad guy and trying to actually ameliorate the effects of that, because that's the cause of the disease. A small number of people are working on the other molecule, the protective molecule, and that's what we've focused on, but we're also connecting it into the sort of bad guy, so we have a sort of a biphasic approach, you might say. So just to kind of summarize kind of where this bad guy beta amyloid, how it actually leads to Alzheimer's, I've taken this picture from a Nature Reviews, a drug discovery article of recent times. So here's the APP I mentioned before, this protein that extends from the membrane, and we had a horizontal last time. Here it is vertical. Here's 2 enzymes that actually cut this at the beta site, this at the gamma site, and they cut out then this fragment, and that's the beta amyloid. Now, if that's produced in low concentration, this will do some good physiological things, we think. But if the concentration gets too high, as shown in this diagram, these things can aggregate. And so here you see 9 of them stuck together. And this is what is the neurotoxic form of this particular peptide. In fact, even if you only have 2 aggregating, or 3 or 4, these are quite toxic species, so you don't need to have as many of this. Interestingly enough, as this builds up, eventually it falls out of solution. This is still soluble in our brain, but it falls out of solution, and we get these insoluble plaques. And Alzheimer's in 1906 was doing post-mortems of brains of people who died having had dementia. And he discovered these plaques in the brain, and that's been a definitive diagnosis for Alzheimer's. For a long time, this was thought to be the sort of cause. We now think of this as just the end product. This is the toxic thing, but the end product of all of this is you get these coming out of solution and forming these plaques. Now, what this also indicates, that there are some places around here that if you're trying to develop therapeutic intervention, that you can focus on. For example, the 2 enzymes that cut this molecule out are potential focus points for therapeutic intervention. This here, if we get this building up, if we can focus drugs on trying to clear this, then that's another potential sort of focus point. And certainly the work that's going on internationally and focus of some of the larger companies are on those two mechanisms. So that's a quick summary of, at the molecular level, what we think is happening in Alzheimer's disease. So I've said that my research lab is focused on the good guy, this other pathway, this other product, which we know is neuroprotective. And so the approach that we took was to try and see if we could mimic what the brain does with cultured cells. So we engineered a human embryonic kidney cell so that it actually produced this good guy protein, the SAPA protein. And it was engineered in such a way as that the protein would be put out into the media which these cells were growing in. And that means we could get a pretty pure protein kind of in one step. And then we could milk the media to collect the protein. So I call this our Fonterra system because it's rather like dairy farming. You kind of milk a cow and then, well, if it's a drought, you only milk it once a day and then you milk it the next day and then you milk it the next day. We do the same principle with our cells on a slightly longer time scale. We milk the media and we keep the cells going and we produce more protein in the next lot of media and so on. And so we're constantly milking this. So here's a result here, an experimental result where we can see what's going on. So here's the cells and days in culture, and we can see at 0 time we don't see anything, and you might just be able to see a light smudge there, and that's an indication that this protein, this is an immunoblot actually, we can just see the protein appearing, and then, so that's at 2 days, at 4 days you can see we've got quite a nice smudge there, which shows there's quite a bit of protein there. Not too much more at 6 days or 8 days. And to get this, and to get it pure in the media, or relatively pure, we actually are starving the cells. So these cells are starving for this period of time. And so what we decided to do was to have them in this starved situation for 3 or 4 days, and then actually give them a bit of a treat to feed them again, give them lots of good nutrients for 24 hours, and then put them back into the starved conditions for 3 or 4 days, and then feed them again, and so just keep cycling. And so on the right here, what we see is the cycles of every 3 or 4 days. So the first cycle it's getting going here, and we produce it in the second cycle, we feed them again, produce it in the third cycle, and we can keep going. And Tate has extended this up to about 8 or 9 times, and the cells keep producing. So this is kind of like why I call it our Frontera system, because we can constantly cycle and actually milk the media for this protein. So it's pretty pure at this stage. It's not perfectly pure, and so we've applied some downstream technology to get it to a pure protein. So we've now got this neuroprotective pure protein produced in the laboratory. So one of the things we wanted to do first then was to check whether what we'd produced was actually neuroprotective, and we've got some assays to actually look at that, again with cells in culture, and it seemed to be great. So it seemed to have this protective quality. So then we wanted to ask some profound questions. Could this protein be put back into memory impaired, compromised laboratory animals and actually do some restoration work? So I'm going to show you that experiment in a minute. And then we asked the question, what about behavioural learning? Could this protein affect how animals sort of learn and remember what they've learnt and apply it? And I'll show you that. And we've also asked, could the protein stimulate the growth of new nerve cells? I'm not going to show you any data for that. That was done in collaboration with Bronwyn Connor's lab in Auckland. But I can tell you that in fact it did stimulate the growth of new nerve cells. So this rather complicated thing here is really reflective of what we think is the best model of mammalian memory. It's an electrophysiological model, and this was done in Professor Cliff Abraham's laboratory. He's a world expert in electrophysiology, and in this particular memory model with live animals. And so, and I want to show you this even though it's complicated, because as a biochemist, having produced a protein in my lab, I was just wildly ecstatic when we were able to take our protein and put it into live animals and get it to do something. And so I'll just take you through what this means. So you can put electrodes into the brains of laboratory animals. They're quite comfortable with this. And you can give them small pulses of electricity and then record what the neurons are doing. And so if we look at this kind of blue trace here, the small pulses are going in and we're recording from the cells here. And okay, there's a bit of a drift, but basically these things keep a relatively stable response, and this is over a 3-hour period. But if a simulation of new information coming into this information processing centre in the brain, area called the hippocampus, occurs, then the cells respond to that, and you can see that instead of just going along this blue line, this is the black line here, they've jumped up. They're sensitized, or what we call potentiated. So this whole process is called long-term potentiation. Long-term because it actually, you can see it's stable, it keeps going, and Professor Abraham has shown that in fact his examples where he can extend this over a year. In other words, those cells will continue in their sensitized state. However, now I want to come up to here, because if we then put an inhibitor, infuse an inhibitor in the brain, which knocks that alpha site, and that allows the production of our good guy protein, then you can see the response of these cells drops off to this here. It's a little bit above the background, but it's quite a big reduction from what they would normally be doing. So these are memory impaired with this inhibitor in there. If we now go to the next one, which is what made me terribly excited. If we now infuse our protein in, the protein that we'd produced in our cells by our Frontera system, if we put that in, infuse that into the brain, this is the green trace, you can see we jump back up to where it was before, and I would say even a little better, but Cliff would probably just temper my enthusiasm on that. And so this protein is able to restore the cells to where they were before. We're able to use this neuroprotective protein then to restore the impaired memory mechanism as determined from this electrophysiological experiment. So that was a very exciting result for me. So we're saying then that the protein has a potential for memory restoration. The experiment I've just shown is it reverses the loss of the natural response of the brain to processing new information. And the next thing we wanted to look at was what about behavioural learning? And this was done in Professor David Bilkey's lab via Professor Abraham. So there are a number of behavioural paradigms for now looking at behavioural tests for looking at how animals respond to various sort of treatments. This is a rather simple one. It's the Morris water maze, and what it is a bath with opaque water, and what you can't see there is there's an underwater platform. So the animal is put in there and swims around, and just like humans do, if there's a platform and a swimming thing, they tend to kind of swim towards that and get on this. These animals tend to do it. So once they know that the platform is there, once they've learnt that it's there, they will actually spend less time in the other quadrants where there's no platform and more time in the quadrant where the bath is. So we set up an experiment where there were groups of animals that were going to have particular treatments, and the first thing you need to do is to make sure they can learn normally. So you go through a normal learning test and make sure all of the animals are learning and can remember. And then you apply your various tests. And so we put with one group of animals the inhibitor infused in. The electrophysiological experiment said that in fact the brain was unable to respond so well to that new information. We also put in our protein along with the inhibitor in another experiment, another group of animals, and the third group, of course, were not affected. So this is a very kind of simplistic conclusion to that experiment. So what hopefully you can see here is there's a red trace which goes basically all over the place, and so the animal is essentially spending equal time in each quadrant. You can now see the hidden platform there, and so this red trace is equivalent to what happened when the inhibitor was put in. So there was a training paradigm. The next day the animals were tested to see if they had remembered, and those that had the inhibitor put in, they couldn't remember, and so they just swam around rather randomly and they would come across the platform. But those that hadn't had anything or that had our protein infused actually, well, they didn't quite make such a beeline, but they spent less time in these other quadrants. They had remembered where the platform was, and they were able to find it relatively quickly. So again, in this behavioural learning paradigm, our infused protein was able to restore to the animals the ability to learn and remember the next day the learning experience they'd had previously. So I was pretty excited about this because it said that, well, this protein might have potential then for development as a therapeutic reagent in humans. It shows restorative brain activity. And then, of course, we have to ask ourselves some key questions. Does it actually have any limitations? Well, actually, it's quite a big molecule. It's a big fragment of the protein. The bad guy is only a small peptide of about 40 or 42 amino acids. This has actually got 612 units. So one of the things we have to do is to try and deconstruct it and find the functional unit, and that's what we've been trying to do with some success, is to find the parts of the molecule which are the active parts. And the idea of this is, can we actually construct then a small molecule which might have a chance of being delivered to the brain because we have to pass the blood-brain barrier. The other question that's important, is there any counterintuitive biology that might suggest this is not a good approach? Well, really, we know that this molecule is neuroprotective, and we've seen these dramatic global outputs, but what we didn't know is actually how it did it. What was the steps by which this was actually functioning like this in the brain? So it wasn't that we had counterintuitive biology, it was just we had unknown biology. So we've been working on trying to fill those steps in. And I want to just give you a snapshot of one of the kinds of experiments we've been doing, and this is the work significantly of Dr. Margaret Ryan, and with one of the other students you saw in the team, Gary Morris, and Joanna Williams, and I have had kind of an overviewing interest in this work. And so here we haven't done this in live animals. We've had to take the same part of the brain, but we've done it with slices of the brain in culture, and what we've done is applied our protein in 3 different times. We've applied it for 15 minutes, or for 2 hours, or for 24 hours, and then what we've analysed is to see whether this affects the expression of the genes. Now, we've got about 20,000 genes, a little bit more. The rat also has about the same number of genes. So we're looking to see whether the expression profile, the activity of those genes changes when we actually apply this molecule, and that's what Margaret was doing. And when you get all of the data from this, it's very complex data, then there are now algorithms available where you can do pathway analysis, and that is to say you're looking at all of the genes that are affected and you try and link them up into specific pathways which will tell you what processes in the brain are being affected. And that's Margaret Ryan is an expert in this kind of analysis, and that's what she did with all of the data. So I'll just show you a couple of pictures from this experiment. So here then is the 15-minute time point, the 2-hour and the 24 hours. And what you're seeing here is red and white, and this is telling you together how many genes were affected. It was about 600 genes out of the 20,000. And most of the genes, that's the red part, were actually what we call upregulated. They had more activity. They were sort of turned on, if you like, and some were actually downregulated. They had less activity. When we went to 2 hours, you can see the pattern changes. In fact, we're now only 40% of those that changed were upregulated. Most of them were downregulated. When we went to 24 hours, that was even more marked, and the number of genes affected was less. So this suggested it was a dynamic process. And when we looked to see whether in fact the same genes were being affected over that time course, shown here by this Venn diagram, in fact most of the gene changes at each time point are unique. They're not shared in common. There's just a small number which actually cross over to another time point. And only 2 genes of those that were affected were affected in all 3 time points. So it's a very dynamic process. We know when we apply then this SAPA molecule, this neuroprotective molecule, there is a dynamic change in gene expression, and that obviously leads through to the kinds of outputs that we saw. So this is what the pathway analysis looks like that Margaret does, and I'm going to actually give you a test in question time just to make sure you've remembered this. The red things here are those that are up-regulated, the green are those that are down-regulated, and there are some that are connected in that may not have showed any change, but they're part of the pathway. So Margaret's derived in different networks at each time, but we can say what these networks mean, and we know this is to do with the remodeling really of chromatin, the DNA, and also gene expression. We know inflammation pathways are important at 2 hours, and then at 24 hours, neurogenesis, the growth of new cells, and it looks like by 24 hours, the brain is sort of trying to actually implement the neuroprotective profile. So looks unintelligible, but actually you can distill out quite a lot of useful information. Okay, so where are we then in 2013 in terms of Alzheimer's disease and treatment? I said at the beginning, the important thing is that really, if we're going to advance in our treatment of this disease, we need early diagnosis. I said that it may be 20 hours before 20 years before the symptoms appear when the disease has started. So we want to push back much earlier to get a predictive or early diagnosis. And so there's lots of international activity around this. Some claims of success. In fact, there was a beautiful paper published in 2007 which looked like it was the team had actually produced a predictive or early that could take Alzheimer's patients and actually select them out. But others that have tried to apply this test have found difficulty. So, there's still work to be done. Even this week, the BBC News reported that a group at the University of Nottingham has derived some diagnostic biomarkers, but then if you read into the story, the head of that group, Professor Morgan, said, we haven't yet validated the markers we've found, and it will be at least 10 years before, if we do validate it, before it would be available for patients. So we have to be a bit careful with some of the claims. Our multidisciplinary team, I haven't talked about this today, is pursuing this, and we're pursuing a new kind of direction, which we think is highly promising. So my conclusion is that it should be possible to get an earlier diagnosis, and I would say relatively soon. What I mean by that is, in the next decade, relatively soon, as distinct from tomorrow or in 6 months. And then in terms of therapies, I said at the beginning, the cupboard is relatively bare. We have a group of drugs affecting acetylcholine degradation, which are mild drugs and somewhat protective. But now the Eli Lilly and Roche, two of the big companies, have drugs in phase 2-3 trials target the enzyme BACE1, and you may remember seeing that on an earlier slide. This is the cutting enzyme which actually cuts at the beta site and is important for the production of the bad guy beta amyloid. And so we know in the Icelandic study, if that cutting is muted, then people that have that state, they actually seem to be protected from dementia illnesses, even when they're quite elderly, well into their 80s. So drugs that target this enzyme, look as though they have quite a lot of promise. So there are major trials just starting on this right now. And also there have been some studies already which have been disappointing, drugs that mobilize the aggregated beta amyloid I mentioned before, but those studies are ongoing. So if you're an optimist, we're in quite a good phase that we should be seeing in the next rather small number of years, I think, some progress on this, and hopefully drugs that are approved by the FDA and are put into the cupboard and made available for people that are suffering from the disease. But the critical thing is that we want to know that the disease is there at an early stage, so there's a chance that these drugs will actually be effective, and it may be if too much damage is done, even though the drugs might be very good, they won't manage to do that. So the wider team, our multidisciplinary team, is grateful for the financial support from these organisations, the Health Research Council of New Zealand, the Lotteries Grants Board, Neurological Foundation and the Marsden Fund, and I thank you for your attention.
Thank you, Professor Tate. I think it's a measure of the man that he was able to impart such technical information in an easily understood conversation, and we thank you so much for that. Are you prepared to take a few questions?
Absolutely.
Okay, so there's a gentleman there. Apologise for the auditorium style.
So if you have these drugs, for instance, these PACE inhibitors, and want to prevent actually Alzheimer's, when do you start using them? So
well,
you can use them for a lifetime, right? So start with 30 and use them for a lifetime.
Absolutely. So, I mean, at the moment, of course, we're not thinking in terms of just giving everybody the inhibitors, but there might be a susceptible group. For example, we know that there's an allele called apolipoprotein E4, which gives some susceptibility for people that have that allele to developing Alzheimer's disease. So if people knew
would you recommend this, for instance, to take these drugs very early on to prevent Alzheimer's?
Well, I'm saying if you had you had the susceptibility allele, then this might be a good subgroup that could actually, take the drugs prophylactically, because it's saying that they've got a greater susceptibility for developing the disease. So, but ideally, we want to have diagnostic biomarkers which allow earlier diagnosis. That's my whole kind of premise here, so that the two things are available, so that if you can show early signs of the disease developing, without the symptoms in a group of people, then you can focus down on those and make the drug treatment available.
Very good question. Thank you. Yes, ma'am.
You have actually touched on it, but an early diagnosis, surely you need a therapy to go with it.
Yes.
Too cruel to be told.
Absolutely.
That you're getting into difficulties here.
Absolutely, and that's, the basic premise I'm saying in our multidisciplinary team is trying to focus on both aspects. And I showed the Huntington's disease, and the reason I showed that, because that's exactly where we are. You can tell people they're going to come down with Huntington's disease at some point in time in their life, just as my family member was told, and she was really waiting for the disease to happen. It's a very difficult situation to be in, and so we don't want to have a predictive or early diagnosis if we don't have anything, in the cupboard that we can do to help the people that actually have that diagnosis. I agree totally.
Thank you for that question. Any more? Yes, sir. Perhaps you wait for the microphone so we can all hear you, sir.
Not all amyloid beta is necessarily a bad guy. that there's more than one type of amyloid beta, and some are actually protective.
Yeah, sure. And
is that your understanding?
yeah, absolutely. I mean, the worst one is the one that's 42 amino acids long. That's because it aggregates more readily than the one that's 40. So we get these peptides that vary a little bit in length. So the one that's 40 doesn't aggregate quite as fast. But I did emphasize that at the right concentration, these beta amyloid is not a bad guy. It seems to be having some normal physiological functions. In the last 2 or 3 years, there's been some paper published, one which suggests it might be acting like an antioxidant for metal ions, to convert metal ions away, like copper 2 to copper 1 as one suggested function. It seems to be acting as a signaling molecule itself, affecting gene expression, certainly affecting the expression of the enzyme which actually produces it. So there's a number of papers in the literature which suggest some positive functions for this molecule at the right concentration.
Just one more question, Professor. Has there been any work done that alongside the genetic predisposition, whether there could be a stealth infection process going on as well?
Whether there
stealth infection? Kind of a hidden infection.
Yeah.
The person might have.
Well, I mean, certainly inflammatory responses is a negative indicator. And, you may be aware that some people have sort of regarded Alzheimer's disease as like type 3 diabetes. So, there are physiological connections with other major illnesses. Now, whether, there's an infectious agent, I think, that's an interesting idea, but without, at the moment, any evidence.
I asked the question a few years back. I read about stealth infections.
Yeah.
And then the more recent reading, for some reason, it just entered my mind.
Yeah, sure.
Whether, there's a connection.
What we know now is that the human body harbours probably thousands of viruses, whereas we didn't kind of appreciate that even a year or two ago. So the potential for things happening to our body physiology as a result of that, of course, is high. And I mentioned with ME/CFS, chronic fatigue syndrome, almost, certainly initiated by a virus and quite profound effects on the brain, so that would be like a stealth infection for that kind of disease.
Okay. You've had, one more from you, huh?
Okay, thank you. You just mentioned that Alzheimer's is a type 3 diabetes type of thing. So is your peptide available also in other types of diabetes, like type 2? Could this be a possibility?
It's interesting that, APP is produced not only in the brain, but it's produced throughout the body. And we don't sort of focus at all on what might be going on in other sort of body systems. So,